Hi everyone, welcome to our Chicken Meat Consortium webinar for today. We have got a very big agenda planned for today, so if we could just ask that all the questions stay till the end, but you are still welcome to use the Q&A function, which is at the bottom of your screen. I'm Rochelle Osmond and I'll be chairing today's session. We will be recording, so the recording will be made available after the webinar today. I also wanted to do acknowledgement of country. So I'd like to acknowledge the traditional owners and their custodians on the lands in which we are all working and present today, and pay our respects to the elders past, present and emerging. Before we started, I also wanted to make note of all of the people and organisations that have contributed significantly to this consortium project. So I'm not going to go through every single one of them, but I have got their logos up there. So I just wanted to say thank you, and just to acknowledge the people and the organisations that are involved in bringing this together. Before we do start properly though, I'd like to hand over to Sarika and Annie from AgriFutures Australia to give a quick introduction. Thank you.
Thanks very much, Rochelle. So good morning or good afternoon, everybody, from depending on where you're joining from. Myself and Annie, we're here today to provide just a bit of an introduction to ourselves, as well as the AgriFutures Chicken Meat Program. So I'll start with the program first. So the AgriFutures Chicken Meat Program, it invests in RD&E or research, development and extension that seeks to grow the long-term prosperity of the Australian chicken meat industry. The program guides investment across 5 priority areas, which are captured in our RD&E plan, which spans from 2022 to 2027. And this plan is what the program uses to invest on behalf of the industry. So as mentioned, there's 5 RD&E priorities that and each has associated planned outcomes, and this is all developed in consultation with industry. So a quick introduction to myself. My name is Sarika Pandya, and I'm one of the 2 research managers at AgriFutures, part of the Chicken Meat Program, and I'm responsible for the priority areas covering chicken health, welfare and biosecurity, as well as efficient and secure chicken production systems, as well as food safety.
Thanks, Sarika. It's great to see so many people here today. Quick introduction to myself. I'm Annie Lane, so I am the other of the 2 research managers for the Chicken Meat Program at AgriFutures. So as Sarika said, we have 5 priorities, and I am responsible for any projects that come under our priorities across environment and sustainability, and also industry workforce. So the consortium project, which is being presented today, sits under our priority number 3, which is contributing to efficient and secure chicken production systems. So this consortium project is largely funded by AgriFutures and represents a collaborative large-scale approach at solving a whole industry problem. So the consortium will be delivered across the life of our current research, development and extension plan. Just to give a bit of a background, our role as program managers for AgriFutures Australia is to really work with our industry stakeholders and researchers to ensure that we're funding projects that are relevant to the industry and also to our research, development and extension plan, and addresses issues and opportunities faced by the industry, and also delivers outcomes that benefit the whole industry. If anyone would like any further information, please feel free to go to the AgriFutures website, and from there you can access our research, development and extension plan, and also further information about the consortium as well. So thanks everyone, and thank you for giving us the time to introduce ourselves, and I'll pass back to Rochelle.
Thanks so much, Sarika and Annie. We will kick off. As I said, we've got a very jam-packed program for you today. So firstly, I'd like to introduce Professor Eugeni Roura. Eugeni is heading up Program 1 and as well as overseeing the whole consortium project. So the Program 1 is on maternal programming and amino acid precision feeding in broiler chickens. So Professor Eugeni Roura, he's a nutritionist by background with specific research interests in digestive physiology and chemosensory science. He joined the University of Queensland in 2010 as a member of the Centre for Nutrition and Food Sciences and also the Centre for Animal Science, which is part of the Queensland Alliance for Agriculture and Food Innovation, also known as QAFI. As I mentioned before, he's currently leading this consortium project. So I will now hand over to Eugeni, excuse me, and let you start with your presentation. Thank you.
Thanks so much, Rochelle. Let me just share my presentation. Can you confirm that you're seeing my...
If we can have it in presentation mode, please, in your notes.
Yes, and I'll swap. Is that correct now?
Yep.
Yeah, thank you.
Thank you.
All right, thanks so much for the introduction, Rochelle, and thanks everyone for attending this webinar. It's a true pleasure to be leading this exciting project. So I do want to pay my respects to the traditional owners and the custodianship of the lands on which we meet. What I'm going to start here before having my two hats, one as director and the other one as leading program one. Let me just very briefly introduce the overall aim objectives of the consortium before getting into the nitty-gritty of program one. As you see in the slide, what I'm trying to summarize in three main points is what this program is about, and it's a nutrition-driven program, mostly looking at key aspects such as reducing the reliance on soybean meal, imported soybean meal, of course, therefore sustainability, while also keep improving performance parameters and particularly feed efficiency. So that's what goes in. We're also looking at the impact on the health of the animal and particularly gut health, and that's what happens inside. So with aspects of our program looking at food safety as well. And finally, but importantly, the impact of all these programs in the environment and societal implications, which will be further explained by two of the leads in program 5 and 6. There was a very ambitious program with 11 priorities that were put together by the AgriFutures Chicken Meat Panel. So the way we organized ourselves was in 6 research programs, and I'm really happy to have all the 6 leaders of those 6 programs today with us, giving us an update on where we are. As you see, program 1 is the one I'm leading, and it's about transgeneration and amino acid feeding, so including breeders. I'll explain a little bit where we are on that program. And then we have program 2 led by Professor Mike Gidley on carbohydrate and amino acid precision feeding. Program 3A led by Sonia and Tim Wester from Massey, well, Sonia from the University of Sydney, sorry, and Tim Wester from Massey University. Then we have program 4 with Dana Stanley. Dana is not here right now, but she has recorded her her contribution. And then we have program 5 on environment, which is led by Mark Dunlop, and we will finish with Professor Ruth Zadoks from Sydney on the integration program. One of the crucial aspects of the consortium is the training of the next generation of nutritionists and researchers in general. At the very minimum, we will be training 10 PhD students, which they are all on board. And we're in fact looking at probably some additional PhD students as well. So that will be a center piece of this consortium. It's a little bit like a training center as well. And of course, as you would imagine, these 10, 12 PhD students will be shared across the different programs. Without further delaying about program 1, let me just jump into the next 5 minutes or so, talking about the research program I am leading. The program I'm leading is about how can we improve broiler breeder nutrition interventions, not only looking at the performance of the breeder itself, but rather focusing in how can we improve embryonic development and the progeny of the chicken progeny, basically. So we have subdivided this program 1 into 4 sub-programs that I will explain one by one. And so program 1 by 1 is truly about feeding broiler breeders and how can we program a stronger broiler, the progeny, through the nutritional intervention in the breeders. I'm not going to explain the 4 years that will be coming. I rather will be focusing on where we are starting, and what you see in your screens is the first experiment, which actually started this last Monday, so that's already ongoing. And what we're trying to do is trying to program, if you want, or programming the biological machinery in the broiler by feeding different programs in the broiler breeder, in the parental, in the maternal diets. So what we're doing is we're going to be looking at 3 different levels of proteins, as you see in the screen, 16, 14, and 12. And one of the hypotheses that we're trying to work out is if we have a low crude protein diet in the broiler breeders, is that going to have an impact on the progeny so that they are more resilient to lower crude protein diets? And we will be doing that by lowering soybean meal in their diets, right? We have a PhD student, Vipul Chandra Ray, and a strong advisory team helping Vipul get things right. The program 1.2 is about in novo interventions, and in fact, we're going to be planning on trying to help embryonic development by supplying additional amino acids in novo, particularly in the amnion. And what that means is that we're going to be looking at using those amino acids which might be limiting in the development not only of the whole embryo, but particularly in the gut. We have a special interest in the gut and the immune system around the gut, the GALT. Particular interest will be looking at the development of the gastrointestinal mucosa, as the graph to the right is representing. We also have 2 PhD students recruited for this program. One is Nadira Di Sanayaka, and Nadira has already started her first experiment where we will be looking at different levels of arginine being the most limiting amino acid in the egg. And so we will be looking at how supplementing arginine to the egg may have an impact on the development of the embryo. We also have Francisca Diaz-Avilés, who has joined the group, and she will be looking at particularly the differentiation of stem cells in the gut and how can we influence the maturation and the migration of these epithelial cells in the early life of the chicken. So program 1.3, it's all about sort of a new term, I guess, which is aminobiotics or amino acids that could potentially be used as prebiotics. Now, David Suiki Chow is the PhD student working in this program, and it's about identifying essential requirements in terms of amino acids of the different bacteria in the gut. So some of the beneficial bacteria, we know they have a high requirement of essential amino acids, while some other detrimental bacteria may not. So how is that impacting fermentation, and how is that reducing the impact on inflammation? That's a good part of what David, together with Connie Tourney and Lida Omareki, will be looking at. The last program, but I do have another slide yet, is about non-essential amino acids. We do know quite a lot about the essential amino acids and the requirements on essential amino acids, but there is still a gap of knowledge on whether non-essential amino acids may or may not be limiting. There's confounding literature around this. So we're going to be looking at the relative importance of non-essential amino acids, particularly in low crude protein diets or low soybean protein diets. Now, the way we're going to do that is by asking the birds, and that's what program 1.4 will be looking at, developing a model where we will be giving choices to the birds, and the birds will be telling us what are the most limiting amino acids. Now, I will be I will be introducing two PhD students here, Anwara Khatun and Bahid Jazi, who just joined the team. And my last slide, if I may, Rochelle, I see you. Sorry for extending this. Is about just the preliminary work that we've done developing the double choice model, basically saying that we have a model that is very robust when it comes to the chickens being able to choose the most balanced diet. In green is a control, no preference. In blue, it's a diet that's deficient in amino acids. And as you see, they are capable at day one post-hatch of selecting the most balanced diet. So that's the principle. And with that, I will lead to the next speaker, Rochelle. Sorry for taking a bit longer.
That's fine. Thanks, Eugenie. Just to everybody that's online too, if you wouldn't mind, when you do put your questions into the Q&A, could you please just say who the question is for? So when we get to the end of the session, we'll know who to direct the questions to. So next up, we have Emeritus Professor Mike Gidley, who was the Director for the Centre of Nutrition and Food Sciences at the University of Queensland. So Professor Gidley's research is focused on structure, function, relationships in biopolymer assemblies such as starch granules and plant cell walls. And this has led to the detailed characterisation of starch and dietary fibre digestion, fermentation in vitro and in vivo, leading to opportunities for optimizing nutritional value of food and feeds. I will hand over to you now, Mike. Thank you very much.
Thank you, Rochelle. Good day, everyone. Good morning, good afternoon. So carrying on from what Eugenie was talking about, I'm going to focus on this program is going to focus on the reality of how the bird gets the carbohydrate and protein from the feed that we give it. And the way that we have characterized the overall aim is about finding sweet spots. So when we feed chickens, then there's the bird which gets nourishment, but there's also the microbiota which gets nourishment. And getting the balance right between nourishing the bird and nourishing the microbiota is one of the key objectives in getting an efficient and resilient avian population. So that sweet spot is about how do we put together ingredients and formulations that provide the digestible carbohydrates and proteins that are critical for rapid growth. So the carbohydrates for the energy, the protein for the amino acids to develop muscles, but also sufficient non-digestible, particularly carbohydrate, that helps to maintain a resilient and robust gut microbiota that can help withstand infection and subsequent potential outbreaks of food poisoning bacteria, for instance. So in terms of the carbohydrate precision feeding, because carbohydrates have this dual purpose that they are the main energy source for both muscle development, and that's primarily the starch in the grains that we feed, and the microbiota, which is primarily the non-digestible parts of grains, which we collectively call fiber. So we've been working in this area for a while in other species, particularly in pigs and in humans. What we're trying to do here is to bring some of those insights with the unique aspects of the avian system to see what improvements we can make in the chicken area. So what we've started off by doing is just trying to understand actually what happens in the digestive system of the animals. So we've been looking at samples that have been provided by Sonia and colleagues from Sydney that have they've got samples from digester at the beginning, the jejunal end of the small intestine, and the end, the ileal end. And this allows us to do a couple of things. One is it allows us to assess what sort of particle size breakdown actually occurs in the gizzard. We understand that the gizzard is an efficient grinding process, and that is why we don't have to grind our feed too much before we give it to the bird, but just what do they actually produce, and what are the consequences of that for the digestion that occurs between the jejunum and the ileum. And so later on, we'll then move on to say, okay, this is what is at the ileum, which is not digested. This is what goes on to the cecum and the large intestine, where the fermentation occurs. So understanding how that transport of nutrients occurs. So just to give you a little bit of data, so what you're seeing here is a particle size distribution just simply obtained by sieving the dry powder from the digester. And what you'll see is there's a very wide range of particle sizes, from things which are very small, less than 50 microns, up to things that are more than 1 millimeter big. So these are particles which are post-gizzard. So the gizzard is a very efficient grinding system to get below 1 millimeter, but is not so great at getting things below 200 microns. And that's, we believe that this is going to give us some opportunities, because if you look at actually what those structures are, you can see these are polarized light micrographs. The multicolored blue and yellow, orange colors things are starch granules. So this is energy. And if you look at some of them, particularly in the wheat cells, wheat samples at the ileum, you can see there's quite a lot of starch still there. It's not available to the bird because it's still wrapped up in the cellular structure of the wheat, because it's still in sufficiently large particles. So we have a target here that we can start to get to grips with, to understand how can we release the maximum amount of energy, because we don't need that starch necessarily to go on to feed the microbiota. There's plenty of fibrous material, which you see in those micrographs, that's available for the microbes, but we need to characterize what that actually is. So what are the outcomes from this? So overall, we're looking to try and develop the formulations and processing tools that will deliver both the digestible carbohydrate and the protein for rapid growth, but also the non-digestible carbohydrate that helps stimulate a healthy gut microbiota. At the same time, if we can develop laboratory screening methods that make it more efficient to screen for new ingredients and to understand how they might work before we start going to in vivo trials. And in order to get the best of both worlds, we want to combine carbohydrate and protein precision feeding with the early gut microbiota establishment, so that we can nourish both the bird and the microbiota, so that the whole system is well nourished and performs to its maximum, and we get the best benefit out of it as eventual users of the animal. So that's it for program 2. I now we'll hand back to Rochelle, but the next talk will be on program 3 on innovative formulations.
Thanks, Mike. Yes, so the innovative diet formulations, that's actually going to be given by 2 people. So we'll firstly start with Associate Professor Sonia Liu. Sonia is a nutritionist that designs practical poultry diets to meet global challenges in food security, sustainability, and environmental change. She's passionate to find solutions for efficient and sustainable poultry production in different production systems. She was awarded the Celestino Beata postdoctoral fellowship just 2 years into her PhD studies at the University of Sydney. Since then, she's worked very closely with scientists from the US, Europe, China, South Africa, and Southeast Asia to develop nutritional strategies for sustainable and resilient poultry production. Thanks, Sonia. I'll hand over to you now.
Thanks, Rochelle, for the introduction. Good day, everyone. Thanks very much for the opportunity to share with you what Program 3A is about. I'm presenting on behalf of my co-investigators and also the whole research team, which I will show their names at the end of my presentation. So Program 3A, there's a strong focus on how we can reduce soybean meal, imported soybean meal inclusion in broiler diet without compromising growth performance. Before I talk about why we care about reducing soybean meal, I would like to talk about the benefit of including soybean meal in our diet. It is important for us to understand soybean meal is a byproduct from soybean oil extraction process, and it is a byproduct from human food and biodiesel industries. Therefore, it is important for us to acknowledge we're already using human industry's byproduct, and that itself contributes to sustainability. We are feeding chickens with the food waste already. However, it's an important issue in South Africa when there is, in South America, when there's an increased demand of soybean farming. However, I just want to emphasize livestock feed, livestock production is not the primary purpose of soybean farming. Then why we still care about reducing soybean meal inclusion? Because in Australia, we purely rely on importation of soybean meal. It is for our supply chain resistance to any change. And that fact we're importing all of our soybean meal lead to very high price in comparison to other regions of the world. And also potentially it increases carbon footprint because we're getting feed far, far away from where we are. There is a strong emphasis of sustainability in our consortium, and sustainability to us in Program 3A, I think it includes the following elements. So the most important thing for us producers and also in terms of sustainability is gross performance and efficiency. This must be maintained when we are manipulating the diet. Any delay of growth, any compromise in feed efficiency means we need more resource, electricity, land, water to grow equivalent meat protein. And we also look into solutions to reduce nitrogen excretion when we are manipulating protein sources in the diet. At the same time, we're hoping this can help to reduce water intake. We're monitoring water intake when we are doing dietary manipulations, and also lead to quality and excreted moisture. So we prefer to use the locally available feed ingredients to see whether this can provide us a solution to completely replace soybean meal by the end of this consortium. And also economical sustainability is very important. And this leads to my next slide. I know at the moment there are lots of talk about reducing soybean meal completely in our diet without considering the cost of gross performance and the feed efficiency. I just want to point it out, it's a very strong societal impact of our research. This is the recent data from Australian Council of Social Services. And in 2022, there are 3.3 million Australians living below the poverty line, which is defined as 50% of national median income. And this equals to $489 a week for individuals and $1,024 per week income for a couple with 2 children. To put this into context, the median rent in Australia is $555 per week. So these individuals and families are going to struggle with food because they can't even afford living by themselves. So it's very important for us to find a solution for the industry, for our consumers, for supermarkets to balance the 3 SDG goals: climate change, zero hunger, and also reduce inequality. So this leads to our approaches, how we're going to do this. We are going to conduct 10 feeding studies at the University of Sydney. And at the moment, we completed the first one. The second one is on its way. Sorry, we're doing the second one, which will be finished before Christmas. And then there are 8 more feeding studies planned for the following 3 years. So we are hoping to provide a short-term, quick solution, which is just focus on local protein-rich alternative ingredients. We can perhaps replace half and 2/3 of soybean meal in a diet. And it's a developing list. The list is, we're consulting the industry partners to see what's relevant to them in terms of alternative protein ingredients, and also how we use feed additives to improve the utilization of all these alternative feed ingredients. Then in the long term, we're hoping also to look into how to use the combination of moderate inclusions of synthetic amino acids and also local protein-rich ingredients, how we can use that to reduce soybean meal inclusion to completely replace soybean meal in our diet without compromising gross performance. Monitoring carcass quality is an important component of this program as well. Therefore, we will be very careful of energy density in the diet to make sure there is no to reduce the fat pad accumulation in the body. So this approach potentially will lead to bird welfare and also flock health benefit on top of the environmental benefit, which most of us are aware. Then another mid and short long-term solution is to focus on minor ingredients and how we can replace soybean meal in the starter phase and in younger birds. And this is also a developing list with consultation from consultation with industry partners. The highlights of our feeding studies, and at the same time, this is also a developing list. We are continuously refining our protocols with consultation from consultation with the industry. So we're using mixed sex birds. They're just a broiler birds. We are no longer using off-sex male birds from the parent line. And this includes both male and female birds. For all our feeding studies, we will gain carcass yield data. We will consult the industry to get practical diets. We want to deliver a solution to you, so this element is very important to us. We will also take some digestibility and maybe plasma amino acid measurement to understand the mechanisms of performance outcomes. And we are collaborating with other programs. With program 1, we are very curious about the amino acid choice feeding studies outcome and how that can inform the feed formulation in our program. And also with program 2, we're very very exciting to learn what's actually left in the diet could not be digested and how that can influence gut health and how we can formulate diet to improve efficiency. And then with program 4 and 6, how feed composition and variation actually influences gut health, especially when we're heading towards antibiotic-free production. Then with program 5, we want to understand how the diet composition change can influence litter quality and also shared environment. So this is just a quick snapshot of the outcomes of the first study. The good news there is from diet 2 to 4, there is no statistical difference in terms of performance, feed conversion ratio, and weight gain. And that's a very good news. So this is all about my presentation, and thanks very much for your attention. Back to you, Rochelle.
Thank you very much, Sonia. Please don't forget to, if you've got any questions, to pop them in the Q&A. But next up, we have our Associate Professor, Tim Wester. So Tim received his PhD in ruminant nutrition from the University of Nebraska in Lincoln before he came to Massey University in New Zealand. His postdoctoral studies were in Houston, Texas, and the Rowett Research Institute in Aberdeen, Scotland. I apologise if I pronounced that incorrectly. His areas of interest since coming to Massey involve basic nutrition across various species, including chickens, cats, and crocodiles, to name a few. Specifically, he studies the effects of feed and processing on nutrient utilization and metabolism. Thanks, Tim. I will hand over to you.
Okay, thanks, Rochelle. I forgot where that introduction came from, but anyway, tēnā koutou, tēnā koutou, tēnā koutou katoa. Greetings from New Zealand. It's a pleasure to be asked to take part in this webinar. Okay, so following up from what Sonia has talked about, we're the second part of this program, looking at innovative diet formulations for modern genotype broiler chickens. So what we're doing, well, first off here, we'll introduce myself, Tim Wester. Also, key to this is Associate Professor Reza Abdullahi, who instigated this, the Massey's involvement with this. He has since gone on to work in France, and he is still involved in this as an adjunct. So he's very much involved with this still. Okay, so what we wanted to do was to look at this, ways to improve sustainability in our growth of broiler chickens. Looking at the footprint of meat production, broiler chickens are actually comes in very favorable in terms of the carbon footprint, and they already are a very good way of producing high quality protein for human consumption. Okay, so what we wanted to do as well is to increase the sustainability by exploring some alternative protein sources that are used in broiler production. You know, we really wanted to reduce this reliance on imported feed. Sonia and Eugenie talked about how soybean meal was not very sustainable because it's not grown in Australia and New Zealand. And we're also at the we're bound to the commodity markets for the price, and so it's very volatile markets. There is also the soybean meal that is used for human diets as well, and also we want to reduce competition of other foods that are used in humans for poultry production also. And by using these local ingredients, again, we're going to reduce the carbon footprint of transportation of some of these feed ingredients. And so we looked at, well, we'll be looking at some of these alternative protein sources. One of them is going to be pulses. That's the study, or the area of our first studies that we're going to be looking at. Another one, we proposed to look at krill meal, but in the interim, this has become unavailable. The supplier is not dealing with this anymore. And then another one, which has been getting a lot of press around the world, is this black soldier fly larva. So again, this is a way that we could increase the sustainability of chicken meat production by not only using ingredients for diet formulation that aren't being used for human foods, but also then using a food ingredient in this black soldier fly that's feeding on food waste as well. And then, you know, we still don't know what else is out there and what is possible and what would be a valuable alternative to look at yet. And so we'll take input on that as well. Okay, so the reason we need to look at our area is going to be looking at determining the nutritional values of these alternative protein sources, and that's so that we can provide accurate information on these ingredients, so that we can formulate diets that have a larger inclusion of these alternative sources and are still meeting those requirements for bird growth. Yeah, and it's important, you know, especially when we're looking at these modern genotype broiler chickens. They're growing so fast, and if you feed a diet that's below those requirements, you end up with a slower growth in those chickens. If you feed a diet that is above the bird's requirements, you end up with wasted feed, and both of these really result in reduced efficiency, which ultimately decreases the sustainability and decreases the profit for the producer. So focus on our progress to date, as opposed to where we're going in the future, because that is also up in the air as well, you know, based on what we're finding. But our progress to date is that we've sourced 5 samples of 5 sources of pulses. These were ground by the University of Sydney and received at Massey. That took a little bit of a process in itself, getting the correct visas to get through and everything. But we've looked at, we'll be looking at chickpeas, faba beans, field peas, lupins, and lentils. And these are all sourced from Australian producers. So we have those ready to go. Before we could get started here, the legislation changed in New Zealand, looking at limiting the housing of birds for production, and so that had a carry-on effect of our welfare committee saying that we needed to renovate our cages and facilities, and so that took a long time to get that underway. As you can see here, we've got nice little cages for them with some artificial grass and some areas for them to perch on as well. We've met that for their we've met the needs for that for the animal ethics, and we have our animal ethics approved for our course of study. And so this works on both the ethics for digestibility trials, so we can measure the nutrient value of these feeds or these ingredients, and then also for performance trials. So when we get good numbers for digestibility and numbers that we can use for formulating these diets, we can test them out and see how the birds respond to them. Okay. The other thing that we needed to do is recruit a PhD student. As you know, as what Eugenie said, that this is a very important area for the consortium, is to train this new people coming through. And so we've got Albright Hamungala coming through. He's a lot friendlier looking in person. I think this was from his visa or his passport application, but he's, a very approachable person. You'll see at the APSS meeting. But he's on board now and just about ready to start his first experiment now. Okay, and speaking about that experiment, we've got, we're going to evaluate these 5 pulses. As I said before, they're field peas, chickpeas, lupins, lentils, and faba beans. And we're going to be determining that nutritive composition of those so that we can provide accurate information, so that when nutritionists and diet formulators go to make these diets using larger inclusions of the field peas and things and the pulses, that they have accurate information on which to balance these diets for. And so our first experiments are going to be looking at finding apparent metabolizable energy values for these ingredients, and then looking at the ileal amino acid digestibility of these as well. Okay. So our treatments for these are going to be, these are just essentially experimental diets, so that we can test for, so that we can measure the apparent metabolizable energy and the ileal digestibility. So essentially what we're doing is we're going to be testing these pulses in either the raw form or in the heat treated form. So we'll be conditioning them at about 80 degrees. And that's to eliminate, well, it'll be to test the presence of anti-nutritional factors on digestibility, and then also to test the effect of heat treating it on those anti-nutritional factors. Okay. So that experiment is just about ready to take place, and we'll have some values for this hopefully in the new year. But that's about it from here. From after we get these values for the AME and the digestibility, then we can look at using these in commercial type diets. Okay. But we need that information on the digestibility and the AME first. Okay. And this is just our team. So we've got me and Reza. We've got a postdoc involved in this as well, Albright, and then our technicians that would be impossible if we didn't have them involved with it. All right. And with that, I'll take it back to Rochelle.
Thank you very much, Tim. Our next presentation is a pre-recorded presentation. Unfortunately, Professor Dana Stanley isn't able to join us at the moment. Hopefully she might be able to join at the end for questions, but she has pre-recorded her presentation. But Professor Stanley has a PhD in molecular microbiology, and she's now a leader in the Molecular Biology and Poultry Health Research Cluster in the Institute for Future Farming Systems at the Central Queensland University in Rockhampton. There, she's focusing on human and livestock intestinal health and nutrition, probiotics, next generation antibiotic development, and pathogen control. Dana is also investigating the ways to reduce and control antimicrobial resistance on farms. So I will now share my screen, and let's hope this works.
We are moving to program 4 now, Precision Early Gut Microbiota Development, and I am Dana Stanley, the leader of program 4 in this AgriFutures Consortium. In this program, we will use the paradigm shift from microbiota, focusing just on taxa, to focusing on function, which is current big trend in gut microbiota and gut role of gut in human and animal health research. So initially we focused just on specific taxa, looking into considering that all lactobacillus, for example, are beneficial, or that all Clostridia or Escherichia species are pathogenic. This is no longer widely accepted approach in gut microbiota analysis. We are focusing on what these microbiota are carrying in terms of function. Do they have any virulence factors, virulence functions, or pathogenicity related functions, such as, for example, toxin production that or invasion of epithelial cells capable enzymes. So we will employ this approach into controlling the first 3 days of colonization. So we will focus on clearing the bad functions from the gut of the chickens, so that bacteria carrying these bad pathogenicity functions do not colonize the birds, at least in first 3 days. Once this healthy microbiota is set up initially, it is much easier for bird to resist colonization of pathogens down the track. So we will use precision-made glycans, to control pathogenic functional groups. These molecules have been shown to be very capable in reducing pathogenicity or targeting. They were developed to target pathogenicity islands and pathogenicity-related functions in bacteria, rather than to target, for example, just cell wall that will reduce or affect all bacteria. So this is the first attempt to aim for specificity of the product. Antibiotics were never specific; they were killing all bacteria. So the approach in designing this product was to aim to make minimum disturbance to microbial community, but targeting simply just probiotic functions. We will use the only currently available precision glycan on the market, but the whole currently there is a big shift on precision designed products, like AI design. This is AI designed sort of product, one of the first ones on the market. I know that lots of major companies are trying to develop not just precision glycans, but we are talking about lots of precision molecules that deliver specific treatments to specific type of cells. So we are talking about exploiting the specificity and precision, and precision is in the name of the product description as well. I am sure that we will soon see lots of similar products on the market. We are currently using the only one available so that we can hopefully get ahead of others in exploiting their ability to specifically interlock with mucus layer and control what comes into the mucus layer, because mucus layer is also glycan, and these precision glycans cross-connect with mucus. So glycans, precision glycan is glycan, mucus is also a glycan structure, and so these carbohydrate chains interlock, and that alters what can enter the mucosal layer and what can digest the mucosal layer based on the preliminary data and lots of published data. So what we want to do is we want to ensure that bacteria that colonizes the chicken gut, those that enter the mucosal layer specifically, are pretty much mostly beneficial bacteria. This is why they come in. This is why we're going to combine precision glycans with known very beneficial lactobacillus probiotic strains to ensure that they come in and they're sealed there in the mucus layer, and then the rest will be also, the remaining parts of the gut, lumen portion of the gut will also be highly influenced by very high load of probiotic strains combined with precision glycans. So we will provide pathogen-free feed. Pathogen-free feed, the feed will be, we will try multiple ways of delivering this probiotic. One of these will be fermented feed that would be offered on a first day in the hatchery. This feed would not be sticky or something. It would be sort of adjusted and optimized sort of size of the particles and palatability and other feed sort of characteristics. So that feed would be really loaded with precision glycan, would be very minor, very low concentration and very high concentration of probiotic strains. So the idea is to control this colonization and influence influence the further progress of colonization in these birds. We know that based on lots of research we did on early gut colonization, we know that we can use this to boost the immune function, not just of the gut, but overall bird immunity, because the colonization process becomes much more controlled. The other thing is that we will be able to use the other aspect of saturating the environment with beneficial microbes, which is done often by spraying probiotics, spraying sheds with probiotics, which is used to control for competitive exclusion of pathogens, saturating the shed with beneficial bacteria to prevent, for example, forming of pathogen biofilms and pathogens, not allowing pathogens to linger in the same shed, which happens when we get recurrent shed in infestations. To just summarize, we have one student already started. She's optimizing lots of things. We have ethics approved. The other student is in medium stage, like his enrollment is okay. We're waiting for the visa. And we are waiting for heat wave to pass through Rockhampton and Central Queensland, which we expect around January and February to actually run the trial, because it becomes the shed temperature, the air conditions and whole climate control struggles once we hit this tropical heat and wet season in Rockhampton. We are hoping to share our first data with you very soon in the next meeting. Thank you very much for your time.
I would like to say that next person up is Dr. Mark Dunlop. Mark is working on the litter management part of the overall consortium. Mark's worked for the Department of Agriculture and Fisheries for over 20 years, and during that time he's mostly focused on poultry production, especially in the areas of litter management, litter amendments, energy efficiency, animal welfare, odour and environmental impacts. Mark's currently working on a project to document the industry's litter management practices, measure ammonia and odour during and after litter conditioning, and compiling ventilation data. He enjoys on-farm research and discussing anything related to chicken meat and egg production with growers and service people. So I'll hand over to you. Thanks, Mark.
Thank you, Rochelle. Share my screen. Looks like we should be live. Yeah, so I'm the leader of Program 5, which is the environmental program, and our primary focus is on litter management.
Can't see your screen.
Oh, hang on.
Mark.
There we go. Sorry, I've forgotten to share the button. There we go.
Thank you.
Yeah, so with litter management, one of the main things we're really trying to focus on is preventing excess water going to the litter. Actually, what we're trying to prevent is to prevent having wet litter. And the ways that we go about this is by preventing excess water going to the litter, increasing evaporation. And another thing we're going to focus on during this project is trying to monitor the in-shed environment and excreta during the nutrition and gut health trials. by the other teams. So one of the things that we know from the past is that there's a pattern to how much water the chickens excrete to the litter. So one of the things that we're going to be doing during this trial is supporting the other programs to understand how to help them to measure the excreted dry matter and moisture going onto the litter, and just to be able to use that in the context of litter management, and applying the ways to assess litter conditions, including using the AgriFutures litter guide, which which you can use to assess the litter moisture and friability during the research trials. So trying to put all this data together so it can be related back to the nutrition and gut health treatments that are being trialled. Another thing that we're going to use to try and help us with this assessment is the use of some new ag tech. There's a robot that's available that's got a lot of capabilities as far as viewing what's happening in the shed and measuring the conditions. And one of its capabilities is to use machine vision to assess excreta quality. So this can be looking for signs of poor gut health or poor or bad reaction to nutrition trials or gut health or whatever's going on. So we want to just use this in the research situation so that we can understand what data it collects and how useful it is in trying to understand nutritional and other treatments as well. The other thing that we're really going to focus on in Program 5 is about trying to increase evaporation rate in shed. So as I said before, we know that there's a pattern to how water goes onto the litter from the birds. And some modelling that we've done previously has shown that there are periods of time where evaporation rate is actually less than the amount of water going onto the floor. And so when the evaporation's less, then obviously the floor will get wet. And this is one thing that we're going to try and address in Program 5. So just sort of going back to basics, the things that we need to do to evaporate water from the litter and to keep it dry is to add heat. We need low relative humidity air. We need to bring moisture to the surface, and increasing the airspeed will increase evaporation from the surface. Industry typically can't do too much about the heat or the controlling relative humidity of the air. As far as bringing water to the surface, a lot of there's already things going on in industry. So, chickens, add their water in their droppings to the surface of the litter, so it's already on the surface. And then through chicken activity and by growers tilling the litter, it tends to bring the moisture back up to the surface. So that stuff's already kind of taken care of. In Program 5, we want to focus on increasing the airspeed or testing the increase in airspeed at the litter surface to see if we can increase the evaporation rate. This concept has already been trialled overseas in the US, and it has really sort of shown that it can be used to reduce the presence of wet litter, and then that reduces foot pad dermatitis and other adverse effects in the shed. The configuration of the fans that they use overseas isn't going to work well here in sheds that have got ceiling baffles, which are still quite common. So what we're going to be doing is trying to turn the fans a different way and have air blowing across the shed rather than along it, but still trying to achieve increased airspeed at the floor. So this is one of our research focuses in Program 5. Another thing we're going to do is continue working on a litter evaporation model that I started in a previous project. We use spreadsheet data. We've used environmental data from real chicken farms. And we've already used this to show the effect of several litter management practices, including tilling, changing temperature and increasing airspeed. But what we'd like to do is to make the model more applicable to multiple growing regions around Australia, so you don't need to use farm-specific data. And then this way we'll be able to assess the effect of these litter management practices under different seasonal and regional effects. And we also know that there's a few little quirks in the model that do things that probably don't really reflect reality. So we need to do a little bit of fine tuning, and we're already starting to collect some new data for it under controlled conditions. That pretty much wraps up what Program 5 is about. So I'll hand back over to Rochelle, and we'll move on to the next one.
Thanks, Mark. Remember to turn my my camera and microphone on before talking again. Okay, so last up we actually have Professor Ruth Zadoks. She's going to be talking about the last program, which is integration. So how all of these programs that we've already heard about, how they're all going to be integrated together. So Ruth is a professor currently in animal production or animal production health at the Sydney University School of Veterinary Science. Her background has been on research into bovine papillomavirus in Zimbabwe, and her PhD work focused on the molecular and mathematical epidemiology of Staphylococcus aureus and Streptococcus urbarus mastitis in dairy herds. She's held a portfolio of projects spanning research into antimicrobial resistance, food safety, Klebsiella and Group B Streptococcus across humans, cattle and aquaculture species in Europe, South America, Sub-Saharan Africa and Southeast Asia. Since joining the University of Sydney, she's focused on interdisciplinary research in the areas of One Health and food security. Thank you, Ruth. I'll hand over to you.
Thank you very much, Rochelle, for that introduction and a bit of a blast from the past. As Rochelle said, my name is Ruth Zadoks. I'm a veterinarian by training, interested particularly in infectious diseases and pathogens across livestock species. And I'm very fortunate to be at the University of Sydney, where I can work with Sonia and her team, and also with the other members of this consortium, because Program 6 is all about integration across the various pieces. And if I can forward my slide, I can show you what we mean by that. So we've heard from Eugeni about essential amino acids. We've heard from Mike about starches and fiber. We've heard from Sonia and Tim about ways to reduce soybean meal in our diets. And we've heard a beautiful recorded presentation from Dana about the microbiota. So fantastic specialized work in each of these areas, but ultimately for our poultry producers, we need to bring it all together. And that's what the program integration is about. Can we pick the best of the essential amino acid work, administration in ovo, for example, and combine that with a microbiome that Dana developed at hatching and see how the two work together. And ultimately bring all of the work packaged together and make them culminate into a practical and affordable and cost-efficient diet. In doing so, we'll obviously look at the environmental impacts in the way that Mark just explained. And then there's one extra component that sits in the program integration, and that's talking about the food safety impacts. And I sort of think of that aspect as the do no harm component of the integration program, and where Sonia has already talked about the Sustainable Development Goals and the importance of zero hunger and climate action. As we modify our diets, we also need to be mindful of the importance of good health and well-being. And that applies both to animal health and to human health. When we think about the animals, things like necrotic enteritis come to mind, caused by Clostridium perfringens. And actually our hope here is rather than avoiding harm, we can actually do benefit and reduce the risk of necrotic enteritis by reducing our reliance on soybean meal. So do no harm is maybe a slightly more negative version than it needs to be. We're also mindful that we need to keep an eye on emerging problems. And I saw a question in the Q&A about myopathies. One of the emerging pathogens we need to worry about is Enterococcus cecorum, which causes back problems more than myopathies. And we don't know a whole lot about how Enterococcus cecorum responds to different diets and different gut microbiota. But the kinky back it creates is a significant problem for the broiler industry. So our consortium work is a good opportunity to gain a better understanding of which strategies might increase or decrease the risk of Enterococcus cecorum causing health problems in our poultry. Of course, we also need to consider the human health or public health side of it. I'm under no illusion that we'll suddenly solve world peace and Campylobacter and Salmonella problems, but as we change our diet ingredients, we do want to monitor what impact they have on Campylobacter and Salmonella. And if Dana is successful with her gut microbiome manipulation, we might actually reduce their levels and also their shedding and their presence in the shed. And it would be wonderful if we could demonstrate that. There are other health concerns that are not spoken about very much in public, but they do exist under the surface. And one of those would be antimicrobial resistance. And some of the work that my colleagues presented might actually have a relationship to that. What you see here is a cross-section of a maize plant. And Mike talked about different starches and starch particle sizes and how the ingredients from those starches might actually reach the gut unlocked. And what you see here, if you can see the colors, is in red, the tissues of the maize plant, and the yellow dots there are bacteria. Some of them are highlighted with a white arrow that you may be able to see. And those bacteria are Klebsiella isolates. And Klebsiella is not necessarily a poultry pathogen, it is a human pathogen. Its importance here might lie in its role as trafficker of drug resistance genes from the environment to clinically important bacteria. We don't know whether that's happening, but it's something that I would like to keep an eye on just to be on the safe side. And likewise with the soldier fly larvae that Tim mentioned, what do those larvae eat? What do they bring to the poultry shed? And hopefully what they bring to the poultry shed is actually an ability to reduce antimicrobial resistance genes and antimicrobial residues. So where I say do no harm, there may actually be an opportunity to demonstrate benefit from the diets and the healthy guts that we're hoping to create. And finally, as you will have noticed from Rachel's bio for me, I'm a bit of an outsider in a poultry consortium. And my role is really to identify opportunities for synergy within, across, and beyond the consortium program, looking at what happens in the poultry industry, but possibly even looking at what happens in other animal production industries, for example, around bedding management and microbiota in bedding. So if anybody has any suggestions for what we could integrate in our program, and I've already seen suggestions about meat quality and about economic calculations in the Q&A. If anybody has any other suggestions about things we could integrate in our consortium's work, we would be very happy to hear it. And with that, I'll go back to our overview slide. And Rachel, I'll stop sharing and hand back to you.
Thank you very much, everybody, for doing a very quick run through of a very large project that's going to take, you know, quite a few years for us to get to the end of it. But we do have some questions, and some of them have already been answered online, but I am going to actually come back to one of them and ask for Tim and Sonia to provide their questions live as well. So the question is the production volume of pulses in Australia and New Zealand enough to cover human food demand and replace soybean meal? From a crop production point of view, which alternatives are more viable to replace imported soybean meal? And have there been breeding programs to reduce anti-nutritional factors of pulses for birds? I'll let you go first, Sonia.
Okay, yeah, thank you. Thanks for bringing this question line, because I'm actually also curious to say whether Mike has any insights. So my very superficial understanding is more and more farmers are using pulses for crop between their primary crops, and this led to lots of pulses on the market at quite friendly prices, and our industries has been using it quite regularly, and that's a very important feedback we received. And the question is how we can use all these combinations of different ingredients we can use locally to completely replace soybean meal.
Thanks, Sonia. I see Mike does want to contribute to the conversation too, so I'll head over to you now, Mike. Thank you.
Okay, thanks, Rochelle and Sonia. Yeah, so the other element in this is the human food element. So a lot of interest in using pulses as a source of protein in human food. And essentially it's the same issue. So what you see in the supermarkets these days as the plant meats, almost all of them are based on soy protein. And so almost all of them are imported raw materials. So that industry is having exactly the same question. Should there be a local source of pulse protein for the human food market? So it really would benefit, I think, the chicken nutrition area to be thinking alongside the foods group as well, because what the food people generally want is enriched protein, and so they take pulses and they fractionate the pulses, and they take enriched protein sources. And the rest is essentially waste, but it's still protein containing. The enrichment they get is not great. So this is an area that we're looking into from the human food point of view, because the viability of any onshore protein, legume protein system really depends on finding profitable uses for all aspects of the legume. My sense is that there's probably as much use for the fraction that is not wanted by the human food manufacturers in the chicken area as there is for the overall protein. So there could be an interesting combination here of uses for Australasian grown pulses, part of it to go to human food and part of it to go to chicken feed. Needs looking at. There is no current industry though, basically. A lot of activity, a lot of willingness to look at it because people see the dollar signs in terms of high plant protein foods for humans. I think this group needs to be on top of that as well and working alongside, because I can see there being some good synergies there.
Thanks, Mike. I think it's a bit of a watch this space then from what you've been said, both of you have said. Tim, did you want to
I think that's no, I think Sonia and Mike have it pretty much hit the nail on the head. Pulses are underutilized for both humans and animals, and it could be a market that could be tapped for us to use, and even using some of those byproducts is what Mike was talking about. I see a lot of proposals coming through looking at these various fractions, but they're using, they're super processing, highly processed foods that they end up with. And so that also calls into question about whether or not they're going to be healthy as well for humans, and a viable food source as well with this highly processed foods. But yeah, I mean, everything's up in the air yet. I mean, there's a lot of different things that we can look at, and these are some of the things that we've identified as well. And in terms of the looking at the selection for the anti-nutritional factors, it's not a simple gene, that controls anti-nutritional factors. It's not something that you can just go in and cut it out. It's tied into, plant defense and, against, because that's what these anti-nutritional factors are for. They're to prevent them being eaten by pests and other animals. And so, it's tied into things like, the growth and the immunology and the other performance traits in it as well. So it's not easy just to cut out that anti-nutritional factor and then still have yield and everything else. So it's not a very easy, simple issue to look at. Okay.
Thanks, Tim. We do have one question for Mark on here. Mark, are you available yet? Is just on litter types. Does litter types affect the evaporation in litter?
Yep, I've just typed an answer, but I can talk about it as well. Different bedding materials will certainly affect evaporation rates, both the different types of material, but also how fine the particles are. So we're talking like shavings versus sawdust. The finer the materials are, the smaller the pore spaces, and they tend to overall slow down the release of water. But we just tried another material recently, and it had a higher evaporation rate than wood pine shaving. So, different materials will definitely also have an effect as well. They had there was a second part to that question about will we be assessing the wind chill effects on young birds? We are planning to put the fans into commercial sheds, and we'll do that under supervision. We still have to do the animal ethics approvals for those kind of trials. If our observations are that the birds aren't affected by it, we'll leave it at that probably. But if there is a sign that we might need to do some more specific research, then we might go back into controlled labs sort of studies to look at the effect of air movement over young birds. One of the things that we're trying to achieve with the circulation fans, though, is to bring the warm air down. So we're not talking about moving cold air, which might be the case for older sheds, but also whenever circulation fans are turned on and off. So we're looking to be using the circulation fans continuously. The research that's been done in the US has kind of shown that there's no negative effect on the young birds. So we're not expecting there to be, but we'll certainly be keeping an eye on it. Thanks, Rochelle.
Thank you. I am mindful of time. We have clicked over the 12 o'clock, but there's just one more last question that I'm going to ask Sonia. Is there an observation on the influence of temperature on the utilisation of nutrients in chickens? Obviously, with the changing climate at the moment, it's an interesting issue when developing feed formulations.
Thank you for that. I was also just typing the answers. Heat stress is such an important issue, and there are a lot of interesting questions. I'm just recently learning this and doing lots of literature research. It is definitely on my personal interest list, but the feedback from industry is quite mixed in terms of whether we should target that as one of the priorities of consortium. But maybe we will continue consulting the industry to see whether this is a priority. At the moment, there is no plan in our experiment designs, everything to test this element, but it is very important.
Thank you very much, Sonia. And with that, I am going to bring this webinar to a close. Again, I want to say a very big thank you to all of the program leads that are part of this consortium. And I know we did cover a lot of ground today, so thank you very much. And we will continue to provide updates as well throughout the next few years. So watch this space. So once again, thank you for joining us, and thank you to our presenters. I do have 2 quick questions at the end of this webinar if you have a chance to answer them. And yes, once we have our recording available online, we will let you know how to access. Thanks again, everyone. Bye.
Thanks, everyone. Thank you.